We're absolutely delighted to have Peter Campbell with us here today. Thank you very much for taking time out of your busy schedule. Peter is originally from New Zealand. He did his haematology training there, as well as in Adelaide, before heading to the UK to do a PhD at the University of Cambridge. He is head of cancer genetics and genomics at the Sanger Institute and has been for a few years now. He's received many awards, for example, the CRUK Future Leaders in Cancer Research Prize. Peter really needs very little introduction. He is a driving force in cancer genomics, and you only have to look at his track record over the last few years. It is absolutely astonishing. So without further ado, welcome Peter.
Thanks, Jane, for that very generous introduction, and it's great to be back at the WEHI. I was last here a few years ago, and at that time I sort of talked about the promise of genomics for unpicking many of the fundamental drivers of cancer development, and I want to present a couple of stories today that take that a little bit further and show some of the complexity of mutational processes in leukemias in particular, and how those mutational processes cooperate to drive malignant transformation. So when we sequence a cancer genome, it's important to realize that that cell that we're sequencing, or that the clone that we're sequencing, can trace a lineage of cell divisions back to the fertilized egg. And during that evolution, those lineages are exposed to a series of mutational processes that drive the acquisition of somatic mutations. And there are intrinsic mutational processes associated with every cell division. There are environmental and lifestyle exposures such as sunbathing and cigarette smoking. Cancers themselves can develop mutated phenotypes that drive the acquisition of mutations, and unfortunately the treatments that we use for cancer are themselves often genotoxic, and we can see the signatures of those processes in the genome. So when we sequence a genome, the set of mutations that we get represent an amalgam of all of these mutational processes that the cell has been exposed to during its history, and we've been one of the sort of more fruitful exercises that we've been engaged in as we scale up the cancer genome sequencing that we've been doing has been to unpick these mutational processes. And it has been, I think, extraordinary how diverse and complicated those mutational processes are across the sort of set of human cancers. So we have lots of mutational processes going on, and they contribute to the development of cancer. From the point of view of understanding the biology of the tumor, we're interested in the mutations that drive the cancer. They're a relatively small fraction of those mutations, probably no more than 1 to 10, maybe up to 20 or 30 mutations within the genome, but the hundreds to tens of thousands of mutations that we see elsewhere in the genome probably are just passenger variants that have come along during the process of clonal evolution. They are a very faithful representation of the mutational processes that are active in the tumor. So we can describe these mutational processes using a series of dimensions, really, which I'm going to kind of illustrate today. We can look at the type of change that we get, substitutions, indels, genomic rearrangements. We can look at where they're occurring in the genome and whether there are specific sequence features nearby those changes that are associated with the increased mutability. We can look at their distribution across the genome, whether they associate with, for example, chromatin state, or they're close to telomeres and so on. We can look for imprints of DNA repair processes. We obviously have our cells have a lot of processes for trying to prevent these mutations, and we can infer the existence of those processes in some of the mutational signatures that we see, and we can look at when they're occurring during that clonal evolution in the cancer, and I'll show you some information about how we can do that. So the disease that I'm going to talk about is childhood acute lymphoblastic leukemia. This is in the case of about 20% of childhood ALLs are associated with a translocation that occurs early in the B-cell ontogeny, which is associated with the creation of a fusion gene, ETV6 ranks 1, from a balanced chromosomal translocation. And the current model for the development of this particular type of leukemia is that this occurs early in the development and stalls B-cell differentiation, and during that, and as that clone expands, there are subsequent cooperating events hitting other cancer genes that lead ultimately to transformation of the cell and disease progression. This event, there's a lot of interesting data suggesting that this initial balanced translocation occurs in utero, and these cooperating events occur over the next 5 to 10 years to produce the leukemia. So as I say, that represents about a quarter of cases of acute lymphoblastic leukemia in childhood. The other disease that I'm going to talk about represents a much smaller slice of ALL, where there's an amplification on chromosome 21 that probably targets RUNX1 and maybe a couple of other genes, and I'll spend some time dwelling on that as well. So just by way of introduction, this is a B-cell malignancy, and of course the sort of major feature of B-cell development is the creation of a functional immunoglobulin gene rearrangement that occurs through a series of programmed physiological deletions of intervening sequence to lead to production of a VDJ rearrangement here, and then that process on exposure to antigen lead with somatic hypermutation, one gets production of a viable antibody. So that process of deletion in normal B-cell development is mediated by a series of proteins called RAG, two proteins called RAGs, which recognize a specific motif that is a heptamer and a nonomer separated by a specific spacer sequence of a specific length. And the RAG proteins recognize this motif and then lead to a cut at that position in the VDJ locus, and then deletion and rearrangement to create the functional immunoglobulin rearrangement. So you know all of this. I don't know why I'm belaboring it, but you'll see why it's relevant as I go through. So for this study, we took 50-odd cases of ETV6 RUNX1 ALL, and we performed low-coverage sequencing for structural variation, and we also performed exomes on 56 of these cases, and called the somatic mutations that were present. And this is the kind of landscape across those 50-odd patients of the structural variations that we can see. And the striking thing here is that the structural variation landscape is dominated by deletions in the burgundy color here. And this is actually a slightly unusual landscape across the various cancers that we've looked at. Deletions are not usually as dominant as this. So, for example, if we look at breast cancer, we see a lot of interchromosomal rearrangements and a lot of tandem duplications. If we look at colorectal cancer, we see sort of large-scale unbalanced translocations and breakage fusion bridge cycles. This particular distribution is quite unusual amongst cancers to see such a predominance of deletions. One of the striking things when we drilled down into these rearrangements was that there was evidence for enrichment of RAG recombination sequences at the breakpoint. So here are a couple of examples. These are canonical immunoglobulin gene rearrangements within some of these leukemias, and as you might expect for the canonical IGH rearrangement, we see clear enrichment at the breakpoint of the heptamer and nonomer sequence, which is what we would expect since that's physiological IGH rearrangement and deletion. But at the somatic rearrangements that were occurring elsewhere in the genome, we also saw some considerable enrichment for these particular motifs. So here, for example, is one of the nonomer motifs and heptamer motifs at a chromosomal rearrangement that's well away from the immunoglobulin locus and so on. And so we saw this occurring much more than we would have expected by chance. And even more enriched were isolated heptamer sequences. So here are a series of structural variations that we see that we saw occurring sort of scattered throughout the genome in these patients. And we saw this motif, this heptamer motif, right adjacent to the breakpoint. This is where the cut occurs, or at least this is where the breakpoint and rejoining occurs. And you can see that the heptamer is showing is very often right approximate to this breakpoint, and always on the 3' end and not on the 5' end of the break. So, in fact, it turned out that around about when we did this analysis, that about 40% of all of the structural variations in these leukemias were mediated by, or at least had evidence for the RAG recombination sequence of either the heptamer or the heptamer plus the nonomer adjacent to the breakpoint, and that is a really striking. It's a really striking feature that there are such a large fraction of these rearrangements were mediated by RAG recombination. One of the classic processes in normal IGH rearrangement is that during the deletion process, there's activity of an enzyme called terminal deoxynucleotide transferase, which adds random bases into the breakpoint junction to further enrich the diversity of the antibody repertoire. And we can see the evidence for this non-templated sequence also being inserted in the aberrant rearrangements occurring elsewhere in the genome in these samples. So here, for example, are the fraction of rearrangements in breast cancer and pancreatic cancer which have either microhomology at the breakpoint, non-templated sequence, or just direct blunt end joins. And you can see it's very similar in that there's a very small fraction, about a sixth of all rearrangements have non-templated sequence in other cancers, but here it was more like 50–60% of the rearrangements had evidence for non-templated sequence being inserted at the breakpoint. Again, evidence that this is at least partly linked to the aberrant activity of the immunoglobulin gene rearrangement architecture machinery. When we looked further, and this has been reported by others as well, from, for example, SNP arrays, we saw really striking clustering of breakpoints across different patients. So here, for example, is a series of deletions which happen to take out P16. And here are 5 rearrangements, deletions that take out P16 within our cohort. And of course, these are under selection. This is a pretty decent leukemia gene. So these are functionally relevant to the leukemia. And you can see that these are all in different patients, these deletions, but they're occurring very closely clustered in space, way more than you would expect. If you looked at a series of P16 deletions in other tumors, of which there are plenty, you would not see this degree of clustering. And we can zoom in on that, and in some cases, they were within a few base pairs of one another, and in some cases even at the same base pair. And interestingly, as you might expect, there was, again, very adjacent the heptamer motif. So this clustering is way more striking than we would have expected by chance, and certainly more than we've seen in other tumors, and again is associated with the activity of these RAG proteins probably. That clustering it may at least partly be due to the fact that this motif is not so prevalent in the genome, but actually, a 7 base pair motif with a little bit of flexibility in the recognition sequence is present many millions of times in the genome, and we wouldn't necessarily expect to see this degree of clustering, even allowing for the fact that these rearrangements may need to be near the heptamer motif. So we looked a little bit more at whether there were other predictors of clustering within the genome, and in fact, really strikingly found that these deletions showed, when we looked at this as the series of possible states, the genome from ENCODE, so basically ENCODE took a lymphoblastoid cell line, took plenty of cell lines, took a lymphoblastoid cell line and did high did chip sequencing for many of the most important histone marks, and then ran a program that basically predicted the kind of state of the epigenetic state of each base within the genome in that cell line, and classified each base into one of these 15 categories. And when we take the positions of the breakpoints, we find that there's enrichment of breakpoints in promoters and enhancers, really striking enrichment, such that in particular deletions which are near these RAG recombination sequences have something like a 20 to 30-fold enrichment in active promoters of genes, that these breakpoints are not being randomly distributed across the genome. And that is considerably different to any other mutational process that we've ever seen. We've never seen such a strong enrichment for particular chromatin states. So there's something about active promoters and enhancers that is drawing this RAG machinery to cause deletions, and we think that that's at least as significant as the recombination sequence for creating this clustering of events that we're seeing in patients. So, as I said, one can look at when these events are occurring, and there's a kind of interest here, because if this is being driven by aberrant activity of the RAG machinery, then it's of interest to know whether that's occurring in a burst. For example, if the patient had a viral infection for which there's been some epidemiological evidence, or whether it's an ongoing mutational process. And one can get at that by looking at, for example, single-cell sequencing. So we took a couple of patients in whom we did single-cell sequencing, and we basically took 100 cells or so from the leukemia, and we measured a series of point mutations and deletions, and these deletions we knew to be RAG-mediated, and placed them on a phylogenetic tree. So this particular deletion here is subclonal, whereas this is found in all of the leukemia cells. And so what we can say that in this particular patient, that there were at least 2 waves of RAG-mediated recombination in this particular tumor. And we saw a very similar thing in the second patient, where there were some RAG-mediated events that were on the trunk of the phylogenetic tree and others that were on the branches of the phylogenetic tree. So at least it looks like it's occurring recurrently as a mutational process. It's not occurring in a single burst. So that just sort of is a quick highlight tour of the properties of this particular mutational process. We see predominantly deletions. We're seeing enrichment of RAG motifs near the breakpoints. We're seeing enrichment of these deletions, really striking enrichment, like nothing we've seen before for any other mutational process in active promoters and enhancers. There's evidence for non-templated sequence at the breakpoint, and it seems to be an ongoing process. So one of the kind of interesting things, when we look at cancer genomes, when we're trying to identify genes which are recurrently mutated in cancer and are biologically relevant for the cancer, the way that we typically do that is to look across hundreds of patients and see if the mutations are occurring more frequently in a particular gene than we would have expected by chance. And that basically relies on an assumption that the mutational process is relatively random within the genome. So if one sees enrichment of mutations in a particular gene, that indicates the process of positive selection. But in this particular mutational process, that's a fairly difficult argument to sustain, because the mutational process is not random in the genome. It's showing clear preference for specific regions of the genome, particularly the genes that are active in B-cell development. So it becomes a harder argument. And in fact, when we look at the levels of recurrence across the cohort of patients here, we can look at whether the deletion we're seeing is hitting a gene that's previously been reported to be important for ALL biology, or is recurrent within our study, or in other known cancer genes. And what we find is that about half of the rearrangements that we see in these leukemias are in genes that have been implicated in ALL biology of some strength. And that is an incredible enrichment. I mean, if you take any other mutational process where the mutations are sort of randomly scattered, well fewer than 1 in 1,000 of those events will be driver mutations. This is a massive enrichment for driver mutations. So somehow this mutational process, because it's specifically targeting genes involved in B-cell development by the fact that it's targeted to active promoters and enhancers, are cutting out the very genes that the cell needs to delete in order to transform. And so it's a very parsimonious mutational process. So this half of the talk basically looking at how we can characterize mutational signatures across many dimensions, and that we really need to understand these mutational processes in order to make our inferences about which genes are cancer genes more accurate. And similarly, that B-ALL seems to be a disease of stalled B-cell differentiation, that many of those transforming events in the leukemia are associated with genes that are involved for the normal differentiation of B-cells. And cutting those genes out seems to stall the cells in that leukemic stage. So the other story, which is quite different in flavor, is of that other subtype of ALL that I mentioned, of amplifications of chromosome 21. And just by way of introduction, so that you can follow the rest of the story, I'm going to introduce two other mutational processes to you, which probably most of you know about. One is breakage fusion bridge repair. And this process was actually first described by Barbara McClintock in Mays back in the 40s and 50s. And basically what she observed in Mays and what has been observed in cancer cells subsequently is that one can get a breakage of a chromosome, probably during G1 or early S phase, and then that break doesn't get repaired before DNA replication. And so what happens then is one gets 2 copies of a chromosome with the same break. And then in the G2M checkpoint, the cell says you can't divide because you've got active open double-strand DNA breaks, and the cell decides that the easiest way to fix these breaks is simply to join the 2 ends together. So now you have a dicentric chromosome with a closely inverted repeat at the site of the original DNA double-stranded break, and then when that cell divides, the 2 centromeres get pulled to opposite ends, cytokinesis happens, and that stretched dicentric chromosome gets cut again, and then this process can repeat in theory, and one can get multiple cycles of this breakage fusion bridge. So this is what one sees at the genome. One sees effectively a copy number change. So one sees more copies of this bit and then fewer copies of this bit. So when we look at the sequencing reads, we see more sequencing reads coming from the bit at higher copy number, and we see these reads reporting a closely inverted repeat at the breakpoint. And this is the sort of thing that you see in a pancreatic. This is in a pancreatic cancer where you can see multiple cycles of breakage fusion bridge. This whole amplicon, including one region of this chromosome 8 that's been amplified up to 24 copies, this whole thing could have evolved in as few as 5 cell cycles. It's a very nasty mutational process that radically restructures the genome and can lead to quite massive amplifications in a very quick period of time. So that's one mutational process. The other mutational process that I'm going to introduce you to is chromothripsis, which is a process, I won't go through the evidence for this, but basically we think what happens is that the chromosome can shatter into probably hundreds of pieces, and then there's a process of DNA repair that tries to put all this basically Humpty Dumpty back together again, but basically winds up randomly stitching fragments together again in this rather haphazard arrangement, and many of those segments get dropped and never get restored into the derivative chromosome. And what one sees in this is a colorectal cancer, what one sees in the genome is this oscillating copy number state, in this case oscillating also between heterozygous and, so retention of heterozygosity and loss of heterozygosity. and this rather chaotic pattern of structural variations that sit across the top of it and basically are linking all of these random retained segments back together in a completely haphazard orientation. We see this process in around about 3 to 5% of cancers, particularly frequent in bone cancers and sarcomas for reasons which I don't think we really understand. There's lots of theories as to how this process is arising and what's driving it, but it's a pretty spectacular mutational process. This is an example in bone cancer where we can see it affecting multiple chromosomes, and again, you can see there's all of these chromosomes which are associated with copy number changes here, all linked together by the series of really weird and wonderful rearrangements. So, coming back to IAMP21 ALL, this was first actually discovered as an entity in a diagnostic lab where in fact the samples that I'm going to describe came from this diagnostic lab where Christine Harrison had been looking for the ETV6 ranks 1 fusion gene that I was describing in the first part of the talk, and basically the way that one diagnoses that in a conventional diagnostic lab is to have a FISH probe that sits on ranks 1 and a FISH probe that sits on ETV6, and you look for them coming together and giving you a fusion signal. And what she found in a few percent of ALLs was that there seemed to be many more copies of ranks 1 and an enriched signal on ranks 1 from that probe, but not particularly fused to ETV6. And she found this, she described this phenomenon about 2 to 3% of ALL. Those children happened to be slightly older than most cases of ALL, and they initially did worse. They had poorer outcomes, but actually the poor outcomes can be rescued with more intensive chemotherapy. And they spent a lot of time mapping the commonly amplified region of chromosome 21, and this is a kind of subset of that data, showing you the kind of copy number profile across the whole of chromosome 21. And you can see there are some kind of interesting features here. There's a common region of amplification around about here, which happens to include ranks 1, but there are a few other genes in this commonly amplified region. There's often this kind of wavy copy number pattern down here, and there's often a terminal deletion at the other end. And in fact, this pattern of progressive amplification and then a sharp drop to a region of deletion is typical of what you would see in breakage fusion bridge cycles. And so they postulated some years ago that this process was driven, that these IAMP21 cases were driven by a process of breakage fusion bridge repair in the patients that they saw it in. So we wanted to demonstrate that was the case, and so we took one of these. We took a few of these cases with sporadic IAMP21 ALL and performed some sequencing to find the rearrangements. Here's one of those cases. You can see the typical sort of pattern of a progressively amplifying region of chromosome 21 of this common amplified region here, and then this is not so convincing because it's right next to the telomere, but there is a very small subtelomeric deletion as well. And when we map the genomic rearrangements in this particular example, we can see evidence for a breakage fusion bridge cycle here, as Christine had predicted, a second breakage fusion bridge cycle here, and then a cluster of rearrangements that occurred in the middle. And I won't go through the details of the bioinformatics about how we did this, but it's possible to show that that the process of rearrangement here had occurred in this particular order. That this event occurred first, this breakage fusion bridge event occurred first, was followed by a breakage fusion bridge cycle here, and then this cluster of rearrangements reflected a chromothripsis event that had occurred after the second breakage fusion bridge cycle. This is a little bit of the evidence for why we think it occurred in that particular order. You can imagine, if we go right back to here, where we have the chromothripsis occurring over 2 copy number states, that's consistent with the straight chromosome being broken and then rejoined. What we're observing here is chromothripsis. The number of copy number states actually was occurring over the number of copy number states within this region of chromothripsis was 3. There were 3 different copy number states. And there are 2 ways that you can get 3 copy number states in a chromothripsis process. One is if the chromothripsis occurs first, and you get a sort of nasty mess of a derivative chromosome, and then there's a subsequent duplication of part of that chromosome, then that gives you a sort of what looks like chromothripsis over 3 states. The other possibility is that the duplication occurs first and is then followed by chromothripsis. In fact, one gets quite different patterns of rearrangements when you do that. So in the chromothripsis followed by the tandem duplication, the bits at the highest copy number state are all linked to one another, as are the bits in the lower copy number state. They're linked to one another, but not across. Whereas here, where the duplication occurs first, followed by chromothripsis, you see rearrangements that join regions at different copy numbers. So this being at 1 being joined to copy number of state 2. So one can actually distinguish which of these processes occurred in which order. So chromothripsis followed by duplication, or duplication followed by chromothripsis. And in fact, when we look at this particular case in the sporadic IAMP21, we see that there are these rearrangements that are joining segments of different copy number, shown as these sort of off-diagonal things here. So what that tells us is that the duplication is occurring before the chromothripsis in this particular case, and that allows us to say that the chromothripsis occurred before the breakage fusion bridge cycles. And we went on to show that by simulations and so on. So this is basically the model then, that you have a normal chromosome 21. In the sporadic IAM21, you see this inverted orientation at the close to the telomeres, and then there's subsequent rearrangement. That's the same thing basically that we're seeing a breakage fusion bridge cycle. In this particular example, we saw a second breakage fusion bridge cycle, and then there was a chromothripsis on the stabilized chromosome, and then that led to the final kind of nasty looking chromosome that transformed the cell. So the other observation that Christine had made during her analysis of these IAMP21 cases is that there was this rather striking enrichment of patients who had a congenital Robertsonian translocation between chromosomes 15 and 21. So for those of you who are not familiar with Robertsonian translocations, they're basically where you have acrocentric chromosomes and the 2, i.e. chromosomes with very short P-arms that get joined together basically at the centromeres. This is a not uncommon, I think it's the commonest chromosomal abnormality in normal people, occurs sort of spontaneously. Of the Robertsonian translocations, actually 1521 is the rarest constitutional Robertsonian translocation. What we found, what Christine found when she went digging, was that about 2% of IAMP21 ALLs had a constitutional 1521 Robertsonian translocation. And whereas the background rate of Robertsonian translocations in the background population is about 10 per million. So the enrichment there is from 10 per million to about 2 or 3%, which means that the relative risk of developing IAMP21 ALL, if you carry a 1521 constitutional Robertsonian translocation, is about 2,500. So a massive, massive enrichment risk for this particular form of leukemia. Now, the striking thing is that as far as we're aware, this particular Robertsonian abnormality does not predispose to any other type of cancer, or indeed any other type of acute lymphoblastic leukemia. It's explicitly associated with IAMP21 ALL. It's not associated with any of the other types of leukemia. And none of the other Robertsonian translocations seem to predispose to IAMP21 ALL. It's only the 1521 that does. So we were obviously fascinated by this incredible enrichment, and asked the question, well, why would a apparently normal child that just happens to have this relatively benign abnormality of the chromosomes have such a risk of this specific subform of ALL. So we sequenced the ALLs, as you might imagine, and this is effectively what we found. So here is a series of what we see in the leukemias is a series of genomic rearrangements that happen to link that go between chromosome 15 and 21, and it looks very much like chromothripsis. And again, chromothripsis occurring over 3 copy number states, and probably chromothripsis involving, well, almost certainly involving the Robertsonian copy of 15 and 21, since we see the rearrangements crossing back and forth across 15 and 21. And when we look using the same kind of analysis that we saw before, we see these same off-diagonal effects here, which tells us that that this chromothripsis is occurring on a duplicated copy of the Robertsonian translocation. So what we think is happening is that there's that during mitosis or during the DNA replication, you get 2 copies, sister copies of the Robertsonian translocation, and we showed that the Robertsonian chromosome is dicentric, and what we think is happening is that that there's aberrant spindle attachment during mitosis on occasion in these cells, and then what that drives is aberrant mitosis, or pulling apart of the chromosomes of this particular chromosome, and it's probably associated with lagging chromosomes, and then we're getting shattering of the two copies of the chromosome, and then those 2 copies are basically being randomly stuck together in a derivative chromosome that then gets, in this case, underwent whole chromosome duplication to give further amplification of the chromosome. So the kind of message, in fact, I don't show this data, but we analysed about 10 cases in total. All of the sporadic, half of them were sporadic IAMP21 ALLs. All of them followed this particular pathway of genomic evolution, where they underwent breakage fusion bridge cycles, followed by chromothripsis, followed by whole chromosome duplication. We analysed about 5 of these Robertsonian 1521 carriers, and in their examples, the IAMP21 ALL involved chromothripsis as the initiating event that occurred on both sister chromatids of the replicated chromosome, followed by 1 or more cycles of whole chromosome duplication. So that's a kind of rather specific series of evolutionary events during the development of these particular cancers, and it's kind of amazing that they occur always in this kind of fixed order. And it's interesting to ask, why is it that particular sequence of events is so necessary for the development of this particular form of leukemia? And of course, the answer is selection, as you might imagine, that the chromosome needs to get to a particular state in order for it to transform the cell. And that sequence of rearrangement events is the most efficient way for the cell to get to that final chromosomal configuration. So just to walk you through the evidence for that, here is basically we have gene expression data and copy number data from a series of IAMP21 ALLs. There's about 40 or 50 of them, plus the rearrangement data, and you can see we can basically average the copy number profile across the genome. So this is the sort of range in green, this is the average in the dark green line, and then a kind of interquartile range in the slightly darker green. So you can see that there's this region of common amplification that's seen in the genome, and as I said, there's this kind of wandering up to the most heavily amplified bit, and then there's often a terminal deletion. That's associated, as you might imagine, with a series of transcriptional consequences that are just driven by changes in gene dosage associated, and so one can see that the most heavily amplified regions of the chromosome have the most elevated gene expression profiles, not surprisingly. Now, one of the things that we can do with the rearrangement data is that we can tease apart the copy number consequences of each of those rearrangement processes. And so And in particular, we're interested in what the effect of chromothripsis is on the copy number profile. And we can isolate that because we know which of those rearrangements are chromothripsis events and which are breakage fusion bridge cycles, etc. And what we see is, not surprisingly, that you get that so chromothripsis is predominantly, it is a deletion process. So what you see is that there's relative sparing of the common amplified region from chromothripsis deletions and a slight paucity of genomic rearrangements in that area, compared with the rest of this chromosome, where there's more clear evidence for deletion, and in particular down this terminal end as well. So that's kind of what you would expect if you were trying to amplify this region. You wouldn't want chromothripsis coming in and removing all of those copies, but it is taking out copies elsewhere on this chromosome. And so we can extract that, and this is basically the average number of copies across the chromosome that are deleted by chromothripsis in the series of cases that we looked at across chromosome 21. And we can ask, what does that look like relative to other cancers? So to do that, we took from a Broad data set where they had SNP array profiles from several thousand cancers of all types, hematological malignancies, solid tumors, sarcomas, etc., all cancers. And what we did was we took all of the copy number changes from all of those thousands of cases and averaged the copy number effects across chromosome 21. And when we layer that on top, we see this remarkable correlation between the aggregate copy number state across all cancers and the effect of the chromothripsis on this in this specific form of ALL. What I think this is telling us is that those the deletion process of chromothripsis is fine-tuning the copy number of chromosome 21 to enhance the transformative potential of those chromosomal rearrangements. So it's not there by accident. It is fine-tuning the selective landscape that this is operating on. So what we're really seeing is a coordinated mutational process that is driving the creation of a chromosome that's optimally poised for leukemic transformation. So this coordinated series of structural variations are exploiting the properties of the different rearrangement processes. So breakage fusion bridge cycles, as I showed you in that pancreatic cancer, that's associated with rapid fire but very focal amplification. You see this really spectacular but quite localized amplification can happen very quickly. Chromothripsis, as I've shown you, gives you discontiguous regions of chromosomal loss. And then I haven't spent so much time talking about whole chromosome duplication, but most of these cases, I think 7 out of the 9 cases had evidence for late whole chromosome duplications. That gives you low amplitude gain of many genes. And you can imagine then that when you stick these together, as we see in the sporadic IAMP21 ALLs, that you get this ability to optimize the dosage of genes across a whole chromosome in quite sophisticated ways. And then you just let selection do the rest. So that's really where I wanted to finish. The other point in all of this is that actually that it's kind of interesting from the basic biology of chromothripsis that we see chromothripsis occurring in the context of breakage fusion bridge cycles, which are associated with dicentric chromosomes, and we see this spectacular enrichment for a chromothripsis-initiated leukemia in carriers of a dicentric Robertsonian translocation. And so there's been some speculation in the literature about the fact that that chromothripsis may be caused by lagging chromosomes getting sort of walled off in micronuclei during the process of mitosis, and this kind of association with dicentric chromosomes is kind of a fuel for that theory. I think quite an interesting thing to explore further, and one of the things we were hoping to do is to generate some iPS cells from carriers of this Robertsonian translocation and see if we can use that to kind of study the process of mitosis in that particular chromosome. It's also kind of the first time that we're aware of that a chromosomal that a constitutional predisposition to cancer is mediated through predisposition to a specific mutational process. And so that's kind of interesting as well. So that's really where I wanted to finish. I'll sort of gallop through mutational processes and how that associates with selection and evolution to drive cancer. Much of the work here, Ellie drove the ETV6 ranks 1 analysis. She's a very talented postdoc in my group, and Yilong is a graduate student bioinformatician in my group who drove the analysis of the IM21 ALLs. And our collaborators for ETV6 ranks 1 was Mel Greaves in London and Christine Harrison for the IM21 in Newcastle. Thank you.
Thank you, Peter, for a very stimulating talk. I'm sure there are plenty of questions.
I'd like to come back to the RAG mediated, or what looks like RAG mediated mutation. So RAG has got a plant homodomain, which not necessarily in RAG, but in other proteins has been shown to bind the histone mark at active promoters, histones be lysine 4-time methylation. So do you think, so firstly, is RAG overexpressed in these patients initially? And secondly, is there any chance that it targets the active promoters?
So we looked at that, and we took apart, I mean, the data I showed you were just on the kind of aggregate chromatin profiles. But when we looked at specific chip sequencing peaks and correlation with breakpoints, it was the trimethylation mark that you're describing that was most strongly associated with the position of those genomic rearrangements. So it's certainly a plausible hypothesis that the RAG protein is binding to that specific histone mark, and that's what's tagging it to these active promoters and enhancers. So I think that seems a very plausible thing. In terms of the expression of RAG proteins, it is known that leukemia cells express RAG proteins. We're not sure why and how. It may just be that it's because the cells are being stalled in that stage of B-cell differentiation that it leads to it being permanently switched on, and it can't differentiate out beyond that where RAG would normally get switched off.
But is there any, I guess it's difficult to tell, but is there any suggestion that these particular patients overexpress RAG?
I think it is hard to tell. We don't know. We've looked in other forms of leukemia for whether this RAG signature is present, and we see it in other forms of leukemia. We see it in the hypodiploid ALLs as well, but we don't see it, for example, in T-cell ALLs. So I think it may well be that. I mean, one of the questions that I wondered about was actually whether ETV6 ranks 1 itself was driving RAG expression, but we haven't really looked properly at that. But I think it's an interesting question about how it is that the RAG proteins are being sort of permanently switched on and active in these cells.
If I can ask a question slightly from left field. So you've provided us a stunning example of the processes of tumour evolution. I was wondering if there's any evidence that these processes might have driven human evolution, like comparing genomes of humans to non-human primates, for instance.
So I think that's a very interesting question. So people have described, for example, chromothripsis occurring in the germline. And in fact, there are, if you look at children born with developmental abnormalities that are associated with chromosomal changes, around about 2 to 4% of those will have what looks like chromothripsis occurring as their sort of to create the genomic changes that cause the developmental abnormalities. There's been, in fact, it's one of those speculations in the literature that dates back hundreds of years. In fact, the term is hopeful monsters. That there's been speculation that during speciation evolution that you might get sudden leaps in genetic change and phenotype that basically that leap across a whole for things that for states that there's no easy path of progressive evolution, that there might be occasional random catastrophic events that suddenly leap over. This is a saltatory evolution is the term that's often used. And I mean, I don't know. I think it's certainly an interesting idea that you might get sort of spectacular genomic change that leads to a sudden shift in genome and phenotype that promotes speciation and evolution. In fact, in some of those constitutional chromosomal catastrophes like chromothripsis, they tend to be quite balanced, and I saw a presentation from someone who'd found an event that had been totally balanced in the mother. It had actually happened in the mother's. The mother was carrying a series of chromothripsis events involving multiple chromosomes, and she was normal. And when she had children, the chromosomes had randomly assorted as they do in meiosis, and her child was born with a whole series of sort of unbalanced chromosomal changes and had the abnormality. So you can actually, it is possible to completely rearrange the genome if it's fully balanced and have a normal phenotype. So it is in theory possible. We don't have much evidence that it really does have speciation.
I've got two slightly related questions about the RAG-mediated deletions. The first is a simple one: is there direct evidence that you're impacting on expression by changing or modifying any of these promoters? And the second one is related to that. I don't know if you talked much about allele frequency of these deletions. Can you see that what you're getting is gradual deletion of these genes associated with differentiation? You get to a perfect point where that clone explodes and you've got very frequent events and those that are subclonal.
Yeah, we found occasional events which just took out promoter, but most of the deletions would take out the promoter, the first exon, and sometimes more, sometimes the whole gene, but mostly the first exon and the promoter. But there were some that were specifically on the promoter. We don't know whether the promoter-specific ones were really having any effect on expression or not. There weren't enough of them to really know. But it's certainly feasible that these events would just take out a regulatory region that would be enough to scupper that gene's expression. In terms of the clonal evolution, actually, Mel Greaves had published a paper a year or two ago looking at this specifically in EDV6-1 ALL, and there it's these leukemias are really are kind of wild subclonally structured. They have lots and lots of examples of parallel evolution. These events are occurring multiple times. In fact, we see that in some of these patients that we would find 5 or 6 different rearrangements hitting the same gene in the same individual, all of which were subclonal when you looked at the copy number changes. So it's a sort of very rich subclonal architecture in these leukemias.
Do kids with trisomy 21 that get ALL, do they still get IAMP21?
Yes, they do. So ALL is increased in Down syndrome quite spectacularly. They do have a slightly increased risk of IAMP21 ALL, but actually more often they have a rather different structure, a different genomic structure that particularly involves rearrangements that affect the CRLF2 gene on the X chromosome. And there are all sorts of reasons why having 3 copies of chromosome 21 somehow interact with that specific rearrangement to drive an ALL process, but I think it's not really been sorted out. It's a very interesting series of observations that Charles Mulligan has made in that space.
Could I ask about breast cancer, of course?
Do you see any chromothripsis in BRCA1 and BRCA2 mutation carriers where you've got defects in DNA replication and repair?
We do. We see chromothripsis across all cancers, and we certainly see it in breast cancer quite frequently. It doesn't seem to be particularly enriched in BRCA1 and 2 compared to wild-type breast cancer. There are probably, I'm not sure why that is. We see other mutational processes in BRCA1 and 2 that we can attribute to the loss of those. So the classic thing that we see are actually small, quite focal deletions of anywhere from a few base pairs up to a few hundred base pairs to a killer base or two. And those deletions, they're kind of interesting. You know, what you see is the loss. You see the deletion, and you see microhomology at the joins, and they're not mediated. They're typical. And they have this sort of broader distribution. If you look at say the set of indels that you get in a HER2 positive tumor or a BRCA wild type tumor, you see a peak of indels at 1 or 2 base pairs with a rapid tail off. What you see in BRCA deficient tumors is a much broader spread of size of deletions ranging from a few base pairs up, and much greater rates of microhomology at the breakpoints. And what that's, I think, is what's happening is that you're getting double-stranded DNA breaks, and the absence of homologous recombination means that the only way that the cell can repair this is using non-homologous joining, so you get a little bit of eating back from the break, and then microhomology-mediated repair. And so we do see distinct signatures of BRCA loss, but not particularly chromothripsis for some reason.
I was really struck, Peter, by your comment that this RAG signature deletion is not seen in TAL. I expected if it was a function of RAG expression. So what's your current thinking about the difference? Is it something to do with some other essential component that is specific to these?
Yes, I think so. I don't know what that is, though. I did, as I said, I have been. I think it seems that this signature is enriched in ETV6 ranks 1 ALL, and it may be that there's something about that environment that leads to aberrant RAG activity. You know, the set of genes that cause TALL are increasingly well described. There's Notch1 mutations, FBXW7 loss, and there are some changes in MIB as well. So we're beginning to piece together the genomic architecture of TALL increasingly well. But seemingly the genomic structure does look different.
So more specifically, is ETV6 in closed chromatin and early T-lineage cells, but in open chromatin in B-lineage?
That's an interesting question that I can't answer, I'm afraid. Yeah, it's an interesting question.
On that note, we should probably draw this to a close. So thank you very much.